The idea that gut microbes influence our eating patterns is now widely accepted. But what if their reach extends further, into the very decisions that shape our relationships, emotions, and risk preferences? Emerging research suggests that the gut-brain axis may play a subtle yet profound role in modulating social behavior, trust, impulsivity, and even moral judgment. The neurobiotic model, which centers on the gut's ability to sense microbial cues and transmit them to the brain through fast neural circuits like neuropods and the vagus nerve, may in fact govern more than hunger and satiety. It may help set the tone of our personalities and guide how we interact with the world. At the heart of this possibility is the gut's role as a sensory and predictive organ. Neuropod cells in the gut lining do not merely respond to nutrients. They also respond to microbial metabolites, inflammatory signals, and immune cues. These signals are relayed in milliseconds to the brainstem, where they influence regions responsible for mood, arousal, and motivation. But the conversation doesn't stop there. These inputs are then integrated in higher-order centers like the amygdala, hippocampus, and prefrontal cortex, regions central to social cognition, emotional regulation, and risk assessment. Several animal studies show that altering the gut microbiota can transform social behavior. Germ-free mice, for example, those raised in sterile environments without any gut microbes, exhibit abnormal social exploration, reduced interest in conspecifics, and heightened stress responses. Introducing specific bacterial strains, such as Bacteroides fragilis or Lactobacillus reuteri, can normalize these behaviors, sometimes even enhancing prosocial tendencies. These changes are often accompanied by shifts in oxytocin and dopamine signaling, both key players in trust, bonding, and reward-based learning. This suggests that microbial byproducts might directly modulate neuromodulator release via vagus-mediated pathways, essentially tuning the brain's social circuitry from the gut outward. Risk-taking and impulsivity may also be shaped by this internal microbial dialogue. SCFAs like butyrate and propionate, produced by microbial fermentation of dietary fiber, have been shown to influence dopaminergic activity in the nucleus accumbens and ventral tegmental area, brain hubs involved in reward sensitivity and impulsive decision-making. A gut rich in diverse, fiber-loving microbes may promote more balanced reward signaling, reducing compulsive behaviors or poor impulse control. On the flip side, a disrupted microbiome, whether due to antibiotics, processed diets, or chronic stress, can dampen microbial metabolite production and lead to overactive or underactive reward responses. This might explain why dysbiosis is often linked with compulsive overeating, addictive tendencies, and even risk-seeking behavior. Anxiety and social withdrawal are also relevant to the neurobiotic framework. Microbial changes are consistently observed in individuals with generalized anxiety disorder, autism spectrum traits, or PTSD. In these cases, altered microbial signals appear to bias the brain toward threat sensitivity, hypervigilance, and emotional rigidity. It's likely that neuropods, sensing changes in the chemical environment of the gut, relay this information to limbic structures like the amygdala, reinforcing behavioral avoidance or mistrust as a protective mechanism. In effect, the brain may read the gut state and assume the world outside is equally threatening or unstable. What's compelling is how fast and adaptive the system may be. Unlike genetic changes or long-term brain plasticity, microbial shifts can occur within days, changing the brain's emotional tone and social style accordingly. A stressful week, a round of antibiotics, or a major dietary shift can recalibrate microbial populations and alter the gut's signaling profile. In turn, this might subtly shift how we evaluate risk, read social cues, or handle emotional feedback, changes that might look like personality quirks on the surface, but are deeply biological at their core. Clinical implications are already emerging. Trials using psychobiotics, probiotics designed to support mental health, have shown promise in reducing anxiety, improving emotional regulation, and increasing feelings of connectedness or empathy. For example, strains like Lactobacillus helveticus and Bifidobacterium longum have been linked to improved social functioning and reduced cortisol levels in stressed individuals. While results are early, they reinforce the idea that the gut microbiome doesn't just help regulate digestion, it may also help regulate who we are. The neurobiotic model expands this even further. If neuropod cells and gut sensory neurons serve as a real-time information stream to the brain, then they may play a role in modulating attention, affect, and trustworthiness judgments in a way that reflects both internal health and environmental safety. A well-functioning gut ecosystem may promote flexible, socially attuned behavior, while a dysbiotic or inflamed gut may skew us toward suspicion, irritability, or social disengagement. In the future, personalized interventions that target these gut-based communication pathways could help recalibrate behavior in conditions like anxiety, impulsivity, or even social phobia. Rather than framing these traits as purely psychological, we may begin to view them as embodied states, expressions of the gut's ongoing conversation with the brain. The neurobiotic sense, in this view, doesn't just help us decide what to eat. It may help us decide who to trust, when to act, and how much of ourselves to share with others. In this light, understanding and caring for the gut microbiome becomes not only a matter of digestive health, but a foundation for emotional intelligence, social harmony, and wise decision-making. The gut may not speak in words, but it speaks clearly, and what it says may help define the very contours of human behavior.